Hello friends and my dear students, this is your Yoganandam biology lecturer from Edall Academy YouTube channel. In this video, we are going to discuss about some tips from the topic biotechnology. The biotechnology unit is one of the most important topic for your NEET examination as well as your board examination. From this topic, you can expect 8 to 12 questions in NEET 2023 examination. Let us discuss one by one. See the first one. Construction of first recombinant DNA done by linking of which gene to plasmid of which bacterium? For the construction of first recombinant DNA, antibiotic resistant gene isolated from E. coli bacterium that is linked to the plasmid of Salmonella typhimurium. So this process carried out by Stanley Cohen and Herbert Boyer in the year of 1972. So this is one of the very important line for NEET 2023 examination. Try to remember what is the name of the gene. Antibiotic resistant gene which is isolated from E. coli bacterium. The second question, tools required for recombinant DNA technology. What are the tools which are used in the field of recombinant DNA technology? Restriction enzymes. So you all know restriction enzymes are broadly classified into two types, endonuclease and exonuclease. And polymerase enzyme, ligases. Ligases are otherwise called as splicing enzymes. It is used to join donor DNA and vector DNA. And vectors, different types of microorganisms are used as gene carriers. Gene carriers are otherwise called as vectors, the host organism. So these are the tools required for recombinant DNA technology. The third point, normal E. coli do not contain resistant against which antibiotics? Try to understand the question. Normal E. coli bacteria do not contain resistance against which antibiotics? So normal E. coli bacteria do not contain resistance against ampicillin, chloramphenicol, tetracycline, and kanamycin. See the next question. Fourth one, why restriction enzyme present in bacteria? Each and every bacterial cell is having at least one kind of restriction enzyme. It is used to protect themselves from the attack of bacteriophages. So here given defense. So it is used to protect themselves from the attack of bacterial viruses, which are otherwise called as bacteriophages. And fifth question, which was the first restriction endonuclease isolated? The first restriction endonuclease is HIND2 isolated from Haemophilus influenzae bacteria. This restriction enzyme have registration sequence GTCGAC. So try to understand, the restriction enzyme can cut DNA at a specific sequence with 6 base pairs. So this Haemophilus influenzae can cut the sequence of GTCGAC. So this restriction endonuclease isolated by the scientist Smith in the year of 1968. See the next important point. The ability to multiply copies of antibiotic resistance gene in E. coli was called. So you all know the multiply copies of antibiotic resistant. Multiplication means that is otherwise called as reproduction. So production of their own kind of another copy is called as cloning. So the ability to multiply copies of antibiotic resistant gene in E. coli was called as cloning. Till now, there are how many restriction enzymes have been isolated? So till now, around 900 restriction enzymes were isolated from 230 strains of bacteria. So this is another important line. Till now, how many restriction enzymes have been isolated? Around 900 different types of restriction enzymes from how many bacterial species? 230 bacterial strains. The next seventh point. The cutting of DNA became possible due to discovery of. So you all know DNA cut into fragments by the using of restriction endonuclease or exonuclease. So after the discovery of a molecular scissors or chemical scalpels, it is always called as endonuclease. So it is used to cut the DNA into fragments. Eighth one, First two restriction enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated in which year? So first two restriction enzymes respond for restricting the growth of bacteriophages were isolated in the year of 1963. Year also important. See the next point. Convention of nomenclature of restriction endonucleases. So here one of the name is given E. coli bacteria represented as ECOR1. That is E. coli. Escherichia coli restriction enzyme. This one isolated from Escherichia coli. E denoted as Escherichia genus name. CO coli species name. R that is the bacterial. That is R mean strain. I it is the Roman number of the following names. This first one mean first enzyme isolated from E. coli. 2 mean that is the second enzyme from isolated from E. coli bacteria, like that. So I Roman numbers following the names indicate the order in which the enzymes were isolated from that bacterial strain. The 10th important point. What is the character of restriction endonucleases? Restriction endonucleases are used to cut each of the two strands of double helix at a specific sites in the sugar phosphate backbone. So restriction endonucleases are used to cut the DNA at a specific sequence. The 11th point. What is palindromic sequence? You all know palindromic sequence mean the DNA sequence of base pairs that reads same on the two strands when orientation of reading is kept in the same. Here one of the example is given. E. coli bacterial. The enzyme which is isolated from E. coli bacteria can cut the DNA at a specific sites with GAATTC. So we are seeing the next strand of DNA that can be we are seeing from backside GAATTC. It is like Malayalam. So you are seeing front and back or back to front. The Malayalam that letters are arranged in the same order, like that. Here one DNA strand is having GAATTC, another DNA strand is having reverse order GAATTC. So when the restriction enzyme can cut the DNA with palindromic sequence is given very clearly in this. This is one of the very, very important question. Cut between G and A on both strands. Then what are sticky and blunt ends? Blunt ends when the restriction site in the middle of the recognition sequence, example ALU, that is 1, SMA 1, HIND 2, SEA 1, all smile in high school. ALU 1, 4 base restriction sequence isolated from Arthrobacter luteus. Here that generic name and a species name is given. Arthrobacter luteus is 1 indicates that is a number. Sticky ends have flanking ends on both strands. Cut is made not in the middle of sequence. Example ECORA, Escherichia coli, RY13, BAM H1, isolated from Bacillus amylolichifacien H. So here some examples are given. Try to understand. These examples are very, very important. See the next 12th one. Enzymes that joins the foreign DNA with vector DNA. So this enzyme is very much used in the field of recombinant DNA technology. Ligases. DNA ligases are otherwise called as splicing enzyme. And the DNA fragments can be separated by which technique? Once a DNA cut into fragments by the using of restriction enzyme, that can be isolated by the using of the technique which is called as gel electroporosis. Gel electroporosis is used to isolate DNA fragments. You all know DNA is negatively charged. So move towards positive charge. See the next 14th one. Gel used in electroporosis is made up of which material? Agarosc obtained from seaweed, which belongs to the family Rhodophyceae, Gracilaria and Gelidium. The agarosc gel is obtained from the seaweed, which belongs to Rhodophyceae, Example, Gelidium and Gracilaria. So these examples are given from plant kingdom. See the next one. In gel electroporosis, DNA is separated on which basis? In gel electroporosis, DNA is separated based on size. Smaller the size nearest to anode, wells on cathode. The 16th point. The process by which separated band on DNA are cut out from gel and extracted from gel piece. The separation of DNA from the gel electroporosis is called as elution. So what is the name of the term which is used? The isolation of DNA, that is separation of bands on DNA are cut out from the gel and extracted from gel piece is called as elution. The 17th point, how bands on gel can be seen? By staining the gel with ethidium bromide. This is another important direct question asked in the NEET examination. So what is the name of the stain which is used to identify the bands on gel? Ethidium bromide, and exposing UV light, the bands on gel can be seen. And importance of selectable markers. So what are the importance of selectable markers? It is used to identifying and elimination of non-transformants. So what is the use of selectable markers? It's used to identifying and elimination of non-transformants. Then 19th one, copy number of linked DNA is controlled by which site in cloning vector? The copy number of linked DNA is controlled by ORIC. Which site? The cloning vector, ORIC. See the 20th one. PBR 322 constructed by Bolivar and Rodriguez. P denoted as plasmid. B represented as scientist name Bolivar or Rodriguez. 322 sequence in which vector is constructed in the laboratory. So PBR 322 constructed by Bolivar and Rodriguez. This is also important. See the next point. 21. Which restriction sites are present in ampicillin resistant gene of PBR322? PST1, Providentia stuarti. PVU1, Proteus vulgaris. So these are the restriction sites are present in ampicillin resistant gene of PBR322. 22. Which restriction site are present in tetracycline resistant gene? The tetracycline resistant gene contain BAMH1 and SAL1 Streptomyces albus present in tetracycline resistant gene. Which site OIN PBR322 codes for protein involved in replication? ROP. Try to understand which site ROP in PBR322 codes for protein involved in replication. See the next point, 24th one. If gene inserted in tetracycline, see the 25th one, in blue-white screening recombinant forms which color colony? In blue-white screening recombinant forms white, that is RAW color. Which color is forming as colonies? White color. In blue-white screening, which enzyme production plays important role in screening? In blue-white screening, beta-galactosidase. What is the name of the enzyme? Beta-galactosidase enzyme production plays important role in screening. The next 27th one, natural genetic engineer. TA plasmid of Agrobacterium tumefaciens can transfer tDNA in host. So it is a natural genetic engineer. 28th one. How to make competent cells for foreign DNA transfer? Calcium chloride treatment with heat shock to make competent cells for foreign DNA transfer. So what is the name of the chemical which is used? Calcium chloride. 29th. Methods to introduce alien DNA into host cell. So there are different methods are used to introduce alien DNA into host cells, such as microinjection, bioelastic or gene gun, It is suitable for plants. And disarmed pathogens, sleep bacterial species, and some viruses can be used. The next point, 30th one. DNA isolation. DNA can be made to precipitate at end by the use of which chemical? So in DNA isolation, DNA can be made to precipitate at the end. Chilled ethanol is used. What is the name of the chemical which is used? Chilled ethanol. What are the main steps of PCR? Here there are certain steps are given. Try to understand. The first step is denaturation. It is carried out 92 to 96 degrees Celsius. It is followed by annealing, carried out 40 to 60 degrees Celsius, and extension requires magnesium ions and DNTPs. It is carried out at 72 degrees Celsius. So these are the steps involved in PCR, polymerase chain reaction. So from this also questions repeatedly asked. See the point number 32. Small chemically synthesized oligonucleotides that are complementary to the regions of DNA are known as. Small chemically synthesized oligonucleotides are primers that are complementary to the regions of DNA. See the 33. DNA polymerase used in PCR is. DNA polymerase used in polymerase chain reaction is Taq DNA polymerase, isolated from Thermus aquaticus. So it belongs to Archaebacteria monera, kingdom monera. So you know Archaebacteria can live in extreme environmental conditions. So what is the name of the bacteria? Thermus aquaticus. From this bacteria only the DNA polymerase isolated and used in polymerase chain reaction. See the 34th one. Protein encoding gene is expressed in heterologous host. It is called. Protein encoding gene is expressed in heterologous host, which is called as recombinant protein. The next 35th one. Equipment required to yield large quantities. That means 100 to 1000 liters culture processing of recombinant product is. So, you know, what is the name of the equipment which is used in the large scale industries? Bioreactors. Bioreactors are the equipments required to yield large quantities of recombinant products. 36.1. After biosynthesis of product, the series of processes done before marketing are known as. So, what is the name of the processing which is carried out? Downstreaming processing includes separation and purification. So this also asked in the examination. Try to remember. The 37th one. Bt toxin makes plants resistant to. Bt toxin makes plants resistant to pests such as lepidopterans, tobacco budworm, orbitworm, coleopterans, beetles, dipterans, flies, and mosquitoes. So this also important point. Bt toxin makes plant to resistant against pests such as lepidopterans, coleopterans, dipterans, and examples are given. Try to understand. And Bt stands for Bt, that is B represented as genus name, T species name, Bacillus thuringiensis. Bacillus thuringiensis. The 39th one. Bt toxin do not kill the bacteria itself. Why? Bt toxin gets activated only in the alkaline pH of insect's gut. So this line also important one. Bt toxin do not kill the bacteria because of Bt toxin activated only in the alkaline pH of insect's gut. Protein encoded by CRY1AC and CRY2AB control which pest? CRY1AC and CRY2AB controls cotton bollworms. Control which pest? Cotton bollworms. The next one, 41. CRY1AB codes for toxin against corn borer. CRY1AB specifically toxic against corn borer. See the point number 42. RNA interference for pest resistance requires double stranded RNA, which gives the complementary RNA for the mRNA to be silenced. So RNA interference for pest resistance requires double stranded RNA, which gives the complementary RNA for the mRNA to be silenced. The point number 43. Insulin chains A and B are linked together by how many inter-disulfide bridges? Insulin chains A and B are linked together with 2 and 1 intra-chain linkage in A chain. 2 and 1 intra-chain linkage in A chain. The point number 44. First recombinant insulin produced. First recombinant insulin produced in the year of 1983 by Eli Lilly and American company. A and chain B produced in E. coli and disulfide bridges created separately. First recombinant insulin produced in the year of 1983 by Eli Lilly. That also asked in the examination. Belongs to which country? American company. See the point number 45. First clinical gene therapy done in the year of 1992. How many years old girl? 4 years old girl child with ADA deficiency. So this line also important. The 46th one. In the patient of ADA deficiency, during treatment, the isolated lymphocytes are genetically engineered using which vector? Retroviral vector carrying the ADA complementary DNA, that is cDNA. ADA cDNA is given. So the patient of ADNA deficiency during treatment, the isolated lymphocytes are genetically engineered using which vector? cDNA. See the 47th one. At present, how many recombinant therapeutics have been approved world over? So at present, around 30 in India, 12 are being marketed. So totally there are how many recombinant therapeutics have been approved? 30 recombinant therapeutics have been approved world over. But in India, so how many therapeutics are marketed? 12. Only 12 are marketed in India. The 48th one. Human protein produced using transgenic animals which is used to treat emphysema. So what is the name of the human protein produced? Alpha antitrypsin. Alpha antitrypsin is a kind of human protein produced using transgenic animals which is used to treat emphysema. First transgenic cow is Rosie. What is the name of the first transgenic cow? Rosie. It gives human protein alpha lactalbumin enriched milk 2 to 4 grams per liter. This also important line. Then flower sour tomato lacks polygalactonase enzyme that digests valpectin. Organization set up in India for ethical issues. GEAC, Genetic Engineering Approval Committee, is an organization set up in India for ethical issues. This is also important line. GEAC, expansion of GEAC, Genetic Engineering Approval Committee. See the 53. Vectors mostly used in Human Genome Project are a Human Genome Project, bacterial artificial chromosomes and yeast artificial chromosomes and host where bacteria and yeast are very much used in the field of Human Genome Project. Large inserts can be used, bacteria and yeast. Thank you very much for listening our videos. Those who are watching our videos, you are requested to like, subscribe and share to your friends. All the best. See the 52nd one. So first one, ampicillin.